All right, so we are at 11 o'clock. Welcome out, everyone, to this webinar for the International Phytobiomes Alliance. I'm Dr. Jason Wallace from the University of Georgia. I'm representing the Microbiome Working Group and will be your moderator today. And before we get into the webinar itself, we've got a little bit of background detail to cover. So going on here, some background on who the Alliance is, if you're not familiar. So the Phytobiomes Alliance is a nonprofit consortium. It's industry, academic, government scientists. And the vision, as it says here, is by 2050 have all farmers, or give all farmers the ability to use predictive, prescriptive analytics based on the conditions of the local area in order to be able to get the best combination of plants and management and inputs, biological or otherwise, in their field. And so essentially it's about plants and everything about it to help with crop production. There are many sponsors of the Phytobiomes Alliance, and we want to say many, many thanks to them because they're the ones that make webinars like this possible. If you don't see your organization on here, you are invited to encourage them to become a sponsor of the Phytobiomes Alliance. And you can contact the Alliance organizers for that for more information. As far as what is a phytobiome, because this is a common question that we are given, a phytobiome is, like I said, kind of plants and everything to do with plants. So on this diagram here, we've got the plants themselves, we've got soil, we've got microorganisms and sort of macrofauna, the little things in the soil. We've also got arthropods, other plants, livestock, larger animals, birds, weather, climate, everything like that. And of course, on top of all of these, we have our management systems, which affect all of these different parts of the system. So essentially, it's a very, very complex system. Now, within the Phytobiomes Alliance, which covers all of that, there are different working groups. This webinar today is being put on by the Microbiomes Working Group, which, as the name implies, works on the microbiome aspect, specifically the plant microbiome. There's also an animal microbiome one, a soil health, controlled environment ag, and then regulations. And they will have other webinars throughout the year that you can watch as well. The Microbiome Working Group specifically has the mission to find the gaps in plant microbiome research and how we can address those to translate into benefits for society. If you want to see sort of our vision of what this looks like, look up this white paper here, From Microbes to Microbiomes. This was published in Phytopathology last year. It's a large effort by the working group and essentially encapsulates our vision of what sort of research needs to happen in this area. As far as future webinars and sessions from the Phytobiome Alliance, so in about a month, we'll have a webinar put on by the Animal Microbiomes Working Group on the ruminant microbiome. And you can see the information and registration here. And then in January, the Alliance will be putting on, again, 2 workshops at Plant and Animal Genome, the conference in San Diego, one on exploring phytobiomes, and then one looking at the uniqueness and commonalities between plant, animal, and soil microbiomes. And so if you're going to be attending PAG, you are welcome to attend those. And in fact, we have a call for abstracts open if you want to submit an abstract for being considered for those workshops. All right, almost done. Little bit of logistics for today's webinar. So this will be recorded. It'll be posted up on the Alliance YouTube channel. And the way it goes is once I finish with this introduction, we will have our 2 speakers, each of whom will have about 20 minutes, and then we will open it up for Q&A. Audience Q&A is being done completely through the Q&A system here. So if you have a question, go ahead and submit it through there. If someone already has asked your question, just upvote it and it will float to the top of the list I see. The chat function is really only for the organizers to talk to you. It doesn't work the other way around. So use the Q&A function to ask any questions you have. And then also after the webinar, there will be the handouts, which are essentially the slide decks for the different presenters. And so that's essential logistics for how it works. So now we get to our actual webinar. So the topic for today's webinar is dissecting microbiome-dependent crop traits from reductionist models to field-scale complexity. And we have 2 outstanding researchers to represent these 2 sides. So our first speaker will be Manuel Kleiner. He is an associate professor at North Carolina State University. He's originally from Germany, where he got his PhD at the Max Planck Institute of Marine Microbiology. His lab's focus is on the metabolism and interactions among microbial communities, both synthetic ones and natural ones. And then I always like getting a fun fact about the speakers. Manuel's fun fact is that he loves reading science fiction, and his favorite author is Alastair Reynolds. So you can also strike a conversation with him about that if you want. Then, once Manuel's done, we will transition to our second speaker, Onyemaechi Henry Obiazikwor. He is originally from Nigeria and is currently a PhD candidate at Murdoch University in Australia. So it is nearly midnight for him. So many props to him for actually coming on this webinar for us. His research focuses on using beneficial microbes to improve crop production in the field. And fun facts about him is that he plays the piano and he loves music, especially gospel music. So with that, I'm now going to cede the webinar to Manuel to start us off. And mind you've got 20 minutes and then we'll transition. Thank you. There we go.
Okay, you can hear me?
Yes.
Perfect. Thank you. Thank you so much. Thank you everyone for joining today. I'm going to talk about understanding key microbial functions related to maize microbe interactions using metaproteomics and synthetic microbial communities. So just to give a very quick rationale for why we're doing what we're doing is, as most of you or all of you might know, plant microbe interactions are really critical for plant fitness and resilience to stress. So all these things that happen in the soil, all these interactions that are ongoing are really, really important for plants to ultimately thrive. Now, unfortunately, it is very difficult to study these microbiome, metabolic, and physiological interactions that ultimately affect the plant phenotype, particularly under field conditions, because it's a very, very complex system. And of course, we can only take snapshots. So what we are ultimately trying to do is to understand how do the metabolisms of microbial and plant species intersect so that they ultimately affect plant fitness. And so one way we're going about this is that we're actually trying to build a comprehensive metabolic model of a maize root bacterial community to kind of be able to predict and model these types of interactions. And so what we're doing is that since it's very difficult to work with natural microbiomes in that regard, we're using synthetic microbial communities that consist of fully tractable microorganisms for which we have complete genomes, which means we can then predict their metabolism and model their metabolism from these genomes and ultimately predict pairwise and community-wise interactions. An additional thing that we're doing, and that's what I'm actually going to talk about today, is we are running experiments where we're using the synthetic microbial communities to then iteratively refine our models to have better predictions. So today I'm really going to focus on actually the type of experiments that we're doing with synthetic microbial communities and metaproteomics to understand how microbes interact with plants. So for this work, we're using a simplified and representative bacterial community of maize, which was initially created by Ben Yu in the lab of Roberto Coldre at Harvard. We've been using this community already for many years in my laboratory now, and it consists of these 7 species. So that's the 7 species that I'll be focusing on today. So the question, or like the experiment I'm going to talk about today, is really addressing the question of what microbial functions are important for root association in maize. And so my graduate student, Anna Gurel, did an experiment where we're trying to tease apart for each individual microbe how it responds to plant colonization. So what we're doing is we're growing the microbes in vitro as well as in planta individually to then compare gene expression responses under these 2 conditions. Now, one thing that Anna had to do was she had to develop a growth medium for the in vitro portion of the work that is very similar to the medium that we're using to grow the plants, so that the responses on the microbial side wouldn't be influenced too much by the medium, but really that we are seeing mostly interactions and responses to root colonization. So she developed growth medium based on the plant growth medium Murdoch, just by adding a few defined ingredients. And if you're interested more in that work, you can find this in this preprint here. So ultimately, she grew all of the microbes individually in vitro as well as in vivo, did, of course, a lot of replication. She did the in vivo portion in this not abiotic plant growth system, where we basically have these bags with a sterile substrate. We use sterilized seeds so that ultimately we really know all the partners that are in our experimental setup. These not abiotic systems are actually something that a lot of people are very interested in the community, because a lot of people are trying to tease apart the interactions of microbes with plants. And for that, we really need to be able to know all the partners in the interactions. And if I have time later today, I'll actually show you briefly our efforts to make better not abiotic systems to improve our experiments. So yeah, so ultimately, she grew these two, she ran the in vitro and in vivo portion. Ultimately, she extracted protein from these experiments, and then we ran an approach that we call metaproteomics, where essentially we're using high resolution mass spectrometry to identify and quantify all proteins in our experiments, to basically get a reading of gene expression at the protein level. And of course, when we do this for the plant root, we also get the full host proteomes, not just the microbial proteomes. But initially, I'm going to focus only on the microbial proteomes. So this is what this looks like now as an example for Herbospirillum robinia. Here you see a volcano plot. Essentially, each symbol in this plot is one protein and its expression level. So essentially the fold change between the in vitro and the in vivo condition. And on the y-axis, we actually have a p-value for significance. So anything that is here colored are actually proteins that are significantly different between the in vivo and the in vitro growth condition. So for this particular bacterium, we identified more than 2,000 proteins, and more than 600 were actually significantly different. That really shows that this microbe really strongly responds in terms of gene expression when it colonizes the plant root. And of course, the functions that are changing are likely involved in interactions or things that relate to plant root colonization. I'll talk about a few examples from this work now, but if you want to know more of the details, we just published a preprint where we have a lot more of the details for this work. So when we look at all 7 SynCom members, the pattern essentially looks always kind of the same. We get very strong changes upon plant root colonization in gene expression for each of the members. And so what does this look like now on a functional level? Here's just an excerpt of some of the functions that we find increasing. So everything in orange is actually in one of these microbes increasing significantly upon plant root colonization. But there's a lot more functions. So if you check out the preprint, you will see those. I'll just briefly talk about 3 of these functions: fatty acid beta oxidation, hemicellulose degradation, and the type 6 secretion system today, just to give you a sense of the type of information we're gaining and what we can do with it. So the first one is actually the biggest head scratcher for us so far, is that we found that fatty acid metabolism is strongly increased in vivo in 3 of the species, and it's particularly the beta oxidation pathway for the degradation of fatty acids that is increased strongly under growth in vivo. And we are actually not quite sure yet what this means. We are not sure where these fatty acids are coming from. They could be coming from wax degradation, plant cell membrane degradation, root exudates, or stress response. So if you have any good ideas of where they might be coming from, or what might be the most relevant source for these microbes that colonize the root, that I would definitely love to hear from you about that. The second type of proteins that we are looking at that are strongly upregulated are type 6 secretion system proteins, which are expressed in 3 of the 7 species. So just as a quick reminder, the type 6 secretion system is a bacterial apparatus that is made out of proteins. It's embedded in the membrane. It's basically a needle-like complex that they can eject and then inject into a target cell. So this needle really penetrates the target cell and releases effectors, and that can have all kinds of effects on the target. So what we found is that in H. robinia, these systems are actually strongly upregulated, while in the other 2 that have them they are significantly downregulated. I'm really now going to focus just on H. robinia as an example, because it has 2 type 6 secretion systems that are upregulated under in vivo conditions. So you see all the different proteins from the type 6 secretion system here, and that you see how expression changes when we are in the in vivo conditions here in orange. So two of the systems are upregulated. And so we're like wondering, is the system directly involved with plant root colonization, or is it more relevant for microbe-microbe interaction? So that's something that we're trying to figure out now. So the first thing that we had to do to be better able to study these type 6 secretion systems in H. robinia is to come up with a way that we can also force or induce expression under in vitro conditions. So Anna and one of her undergraduate students did an experiment where they tested a lot of different media conditions and other conditions, and to see if we could induce both of these clusters of type 6 secretion systems. And when it's here in yellow or bright orange, then that means it's strongly upregulated under specific conditions. So she found that for one of the systems, it can actually be induced when the microbe is grown on solid media, so on triptych soil agar plates. For the other one, it's actually strongly induced under stationary phase conditions. So it potentially has to do with nutrient limitation. So we now have the ability to in vitro induce these systems so we can study them more. Another thing that we've been doing to kind of follow up on this work is we have been working with the group of Nathan Crook, who is a microbiome engineer, and they have been able to generate genetic tools for 5 of these 7 microbes, so they can now transform them and make mutants. And so they have created knockout mutants for us where they knocked out one of each of the 2 systems or both of the systems together. And so here you see growth of Herbaspirillum on corn roots, and what we are essentially counting is colony forming units per gram of fresh weight root. And you see that colonization actually strongly drops or significantly drops when we knock out both type 6 secretion systems. So this is some initial evidence that the system really is relevant for colonization of the plant root. And we're following up now with other work where we're looking at also microbe-microbe interactions. The final little vignette I wanted to give you on what genes are changing is in regards to hemicellulose degradation, which is maybe not completely unexpected, with increased strongly in vivo for Herbaspirillum robinia and Cortobacterium pusillum. So we have these 2 bacteria, and they have genes fuchsilin degradation and arabinoxylin degradation, although missing some of them in H. robinia, and they're strongly upregulated in vivo, so indicating that they're using hemicellulose as a carbon source under in vivo conditions. And so we haven't created knockout mutants for this particular set of genes yet, but we did a follow-up experiment where we in vitro checked if these bacteria can grow on hemicellulose. And so here you see an example of optical densities of growth of Cortobacterium over time on a simplified glucose malate medium, and we get good growth, no carbon source or silane as the carbon source. And you can see that we do get good growth on silane after 24 hours. So it clearly can use a silane as a growth substrate. One thing that I'm unfortunately not going to be able to dive into today is the fact that when we do these metaproteomes of root samples, we're actually not only identifying and quantifying microbial proteins, we're also identifying and quantifying a lot, many thousands of host proteins. And so here you see a PCA plot where we are clustering samples, so each symbol is a sample, by host gene expression. And you can see that the plant seems to be very strongly responding to inoculation with different microbes. So clearly different microbes induce very different gene expression patterns in the plant, and that's something we're going to follow up in the future. So the next steps for our modeling effort is to now integrate our experimental data with the metabolic models, which are essentially a mathematical description of the genetic potential of these microbes, and then that gives us stoichiometry and identified metabolic reactions. So we'll integrate the experimental data. That ultimately we want to go for models that not only do individual organisms, but actually are a model that includes the interactions between multiple organisms, including the plant. And then we can do analysis of fluxes in these microbes and try to see what parameters have to be changed to, for example, optimize growth or things like that. And so with this, ultimately, we'll have a good way to predict for future experiments and test different types of hypotheses in silico, which can then also be tested in vivo or in vitro. To sum up this part of my talk, and I have a few additional slides right after this to show a few other things. So we developed a microbial growth medium to study microbial gene expression in vitro and in planta under very similar conditions. And that ultimately enabled us, together with metaproteomics, to identify hundreds of genes and functions that are upregulated by bacteria when they are growing in planta. So it's a good way to discover these genes. And finally, as a next step, we will integrate this experimental data with genome-based metabolic models of the seven-member synthetic community to make predictions and develop further experiments. So what I wanted to briefly mention today also is these not-abiotic systems, right? I mentioned that not-abiotic systems are very, very important to start teasing apart the individual interactions between plants and microbes, where we're really growing plants under known sterile conditions or with specific microbes. And a lot of members in the community are currently trying to develop this system, and we're all kind of trying to reinvent the wheel. I was quite surprised when I discovered that in the 1970s, there was already a review article on people developing these systems and using them. So we've tried a lot of things because ultimately we want to develop systems that are high throughput so we can work with a lot of plants. And this is all work done by my graduate student, Clara Tang. We want to be able to grow plants to larger stages because, for example, the bags are very limiting. We want to be able to irrigate and drain so that we can do drought experiments and also, of course, improve our growth substrate. So we have done a lot of work. I'm not going to talk you through all the different failures we've had over the years. But I wanted to show you, because I'm very proud of this, what we've come up, what is our current working model. So we have come up with a system that has very large cylinders that contain a growth substrate, and all of this is sterilizable. We have very large glass cylinders that allow for plants to grow to very large size. This system actually has drainage and also an irrigation tube, so we can keep this under fully not abiotic condition while adding water and also water being drained out, so we can use this for drought experiments. So we're very excited, and we hope to get us out to the community in the near future. Here's just a quick glimpse on what growth of corn looks in the system, where we're comparing growth of corn in our previous bag system versus our new cylinder system. And you can see the plants that were grown for the same length, they look very different, right? The plants in the cylinder system are much, much more vigorous and healthy and bigger. So the system is really, like, I think, a big step forward in working with not-abiotic systems. The final thing I wanted to briefly mention, because I think it might also be of interest to the community, we've been working with a lot of researchers globally to create a maize root isolate collection, where we very carefully curated the strains that are in the collection to try to get a really good coverage of the bacteria that are found in corn roots. We're planning to expand this later to the phylosphere and fungi and other things. But right now we've been focusing on bacteria for the root. We have currently 88 isolates that cover a lot of diversity. We have deposited all of this to the German type culture collection, DSMC. So these strains will be available for them from there for everyone to use. They're going to have like some special system where you can purchase strains in bulk. So it's going to be cheaper, and it's going to be available before the end of 2025. So if you're interested in that, keep an eye out for that, and I'm happy to share information about it. And with that, I would like to thank my awesome team here at NC State. I already highlighted a few people that have been driving this research in particular, and also our collaborators that have been involved in the work and our funding sources.
All right, thank you, Manuel. So we are now going to transition to the other side of things. Our goal with this webinar was to show the two halves of microbiome research, the very molecular lab work and then also the field work, because the plant microbiome spans all of that. So now we're going to transition to the other half of that, to Henry. So Henry, if you can please share your slides when you're ready. And we will do the Q&A at the end, answering questions.
All right, thank you. My talk is looking at the potential biological products if they can promote tomato growth in the field and also induce changes to the microbiome to reduce root knot nematode infestation. This is actually part of my PhD work. right? As a way of introduction, the rhizosphere microbiome can protect crop plants, including tomatoes, against stresses. Actually, is the first line of defense against biotic stressors, some borne biotic stressors. So the rhizosphere is the soil zone that is closely attached to the roots, and under the influence of root exudates, then can we now manipulate the microbiome, the rhizosphere microbiome, for the host plant benefits? And the answer is yes. And a good candidate that we can use is biological products. Biological products are agricultural additives that are derived from living organisms. They could be derived from plants, animals, microbes, fish, and seaweeds. So they have a great potential to be used to manipulate the rhizosphere microbiome to increase crop productivity. And also they are good candidates that we can use as alternative to synthetic fertilizers. Here I was trying to look for evidences of the rhizosphere microbiome to protect plant against soil-borne biotic stressors. So I looked at how the microbiome of healthy and infested plants were different, and I found that they were really different. The healthy plant had more healthy microbiome in terms of abundant beneficial microbes and also complex co-occurrence networks compared to the infested ones. And I found that the microbiome actually used different mechanisms to protect plants against soil-borne biotic stressors, including competition, antagonism. Yeah, but overall, there were very limited feed-based studies looking at the rhizosphere microbiome of field-grown tomato crops and how we can apply biological products to improve the microbiome. And most of the studies actually focus on bacterial weeds, which means there's need to also test other important soil-borne pathogens, including root knot nematodes. So for this work, I looked at if biological products can promote tomato growth and induce changes to the microbiome to reduce root knot nematode infestation. And there were two objectives. So for the first objective, the hypothesis was that biological products will alter soil physiochemical properties and change bacterial community composition and or abundance, promoting tomato growth and yield. So this is an overview of the method. Field trials were conducted. Actually, 4 field trials at 2 sites or 2 commercial farms. Farm A was a conventional farm where the products were applied in addition to synthetic fertilizers, while Farm B had just the products applied. And I used 3 commercially available biological products, which were microbial, organic acid, and fish plus kelp or seaweed products. This is like general name for these products. I'm not going to mention the brand names for confidential purpose, but if you want more information, you can reach me and then I can direct it to the appropriate bodies. Then I measured plant growth, including biomass and yield, then soil physiochemical properties, and also I looked at the microbiome, looking at bacteria and fungi in the rhizosphere and box soil. So for the microbiome, I targeted specific gene regions, including ITS for fungi and cystinus for bacteria. So I'm going to just show some of the results from these field trials. As is a big study, I'll show only a few results. So this is the yield from Farm A for trial 1, which is one of the season 2022-2023. The x-axis is showing the treatments, while the y-axis is showing the yield and kg per plot. We can see a little bit of variation there, at least for the microbial product, there was 4% yield increase, but this was not statistically significant. But if you think of a commercial scale and that it has direct practical application, it could be significant. Then in the second season, which is trial 2, I also found more than 7% yield increase for all the biological products that were applied compared to the control. Then at Farm B, which is a non-conventional farm, there were also increases in yield with the products that were applied compared to the control. in the first season, which is trial 1. Then in the second season, also more than 6% yield increase for all the biological products applied compared to the control. You can see there were variation between the seasons, which also pinpoints the complexity that could be associated with feed trials. So looking at the bacterial community composition at Farm A, I wanted to see how the communities in the bulk soil and the rhizosphere, how they compare, and also the trials between the 2 seasons, how are the bacterial communities, and also the influence of treatments. So on this slide, each of the shapes or the dots you see represent the bacterial community of one replicate sample. So something was clear that the trial was actually the most influencing factor on the bacterial communities, explaining up to 30% of the variation. And this was consistent all through, both for fungal communities at Farm A and also bacterial and fungal communities at Farm B, as I'll show in the subsequent slides. So this is for fungal community composition at Farm A. So the 2 trials were still different, and 42% of variation was explained by this difference. So Farm B also had this similar trend with bacterial community composition different from the 2 trials, and also for fungal communities. And this could just be due to the fact that the 2 trials were different in the 2 seasons, because they were planted in different plots within the farm, and also it could be because of seasonal variation. And overall, I did not find any significant treatment effect on the bacterial community or fungal community composition. We just suggest that management practices or even long-term legacy effect of soil physiochemical properties is actually stronger than just applying products for 4 months, may not be substantial to change bacterial or fungal community composition. But however, looking at the abundance of the bacterial and fungal communities, here is showing bacterial communities. First, there were kind of slight differences with respect to the treatment. I will show especially for fungi. We can see the dominant tarsals also were different from the 2 trials. We can see rhodotorula was higher for microbial and organic acid product compared to the control. And this has been known to have beneficial properties, like it can be used as biological control agents. So similarly with Farm B, looking at the bacterial communities, I found that the dominant tarsals were also different with respect to the trials. Different seasons have their different dominant tarsals. Just because, before I forget to mention that the 1.5 you see here is actually all the communities that had median relative abundance of less than 1.5 were aggregated together for ease of visualization. So you can see Planococcus was dominant in trial 2, but in trial 1, Micrococcus was the most dominant. And you can also see Vicina bacteriaceae was higher in plots that receive the organic acid product compared to the control. Then for fungi, I found something very interesting with respect to the treatments. I found Motrella to be higher in organic acid, fish and seaweed treated plots compared to the control. And this was consistent for both seasons, trial 1 and trial 2, even though they were conducted in different plots. So in an attempt to understand why these differences between the trials in terms of the bacterial and fungal community composition, I looked at what soil factors are actually driving these differences. Then overall, I found that the different sets of environmental factors were actually driving the communities. So for example, you see ammonium nitrate was dominant or significant only in trial 1, but not in trial 2. We just suggest that this could be one of the explanations why the 2 communities in the two trials were different. And this was true for fungi also, and both farms in all the trials. So from these feed trials, we can see that indeed biological products can promote tomato growth and could be a good alternative to be used as synthetic fertilizers, even though community composition did not change. Following the application of these products, I found the abundance of some taza to increase, but then I cannot pinpoint, say exactly this is the taza that is responsible for yield increase. So they still need to validate the finding in terms of isolations of beneficial microbes. So you can see clearly where my work is complementary to Manuel's presentation. So we still need to isolate microbes and probably test the microbes in more controlled conditions to validate what they can do. Then long-term application of biological products to the same plots may be more beneficial and have more substantial impact on the microbiome. For example, applying the products to the same plots, or rather to all plants grown on the same plot. For example, if I plant watermelon this season, apply the product, then plant tomato in the next season, apply same products to the same plots, and long-term application, it's expected to have greater impacts on the microbiome, increasing crop productivity sustainably. So next, I wanted to see, okay, the products that were applied in the feed, can it precondition the soil to reduce root knot nematode infestation? So the hypothesis here is that biological products will precondition the soil to reduce nematode infestation by inducing changes in microbial communities, specifically bacterial communities. So because I cannot inoculate the plants in the feed, so I had to collect the feed soil to do the experiment in the greenhouse, but the biological products were applied in the feed. So I collected the soil in the second season after harvest from one of the farm, which is the non-conventional farm. Then did a root knot nematode assessment of the soil, and there was no nematode detected in any of the soil samples, at least using whitehead tree method that I used. Then I mixed the soil with the river sand to create that conducive environment for the nematode to thrive. Then I had this setup where I had controlled microbial organic acid and fish and seaweed. Also, I had inoculated and non-inoculated group. So for the inoculated, they were inoculated with Meloid Ogoini javanica, which is a species of root knot nematodes. Then for the measurements, I looked at bacteria communities, both in the rhizosphere and roots. Then I scored tomato plants for symptoms. I scored the roots, then also extracted eggs from infested roots and counted. And the whole of this greenhouse experiment was repeated for reproducibility of results, and all results were combined as there was no difference between the two trials, repeated trials, except for egg count. So I'm going to show some of the results here. So this is showing root knot nematode symptom scoring. The x-axis is showing the treatments or the biological products that were applied in the feed, while the y-axis is showing the symptom rating. So we can see a great significant reduction in symptom level. Even though the products were applied in the feed, there was no subsequent application done in the greenhouse. That is very interesting, up to 25% reduction in symptom rating. In fact, for the microbial products, there was up to 30% reduction. Then looking at the egg count, as I said earlier, the two trials that were repeated, the egg counts were different, so I presented the results separately. But what is clear that the effect of the treatment was consistent. So there was reduction in egg count, significant reduction in both trials for plants that were grown in feed soil that had products applied compared to the control. Then for bacterial alpha diversity and community composition in the rhizosphere and roots, you can see there was a clear difference in alpha diversity based on Shannon index. So diversity was significantly lower in the roots compared to the rhizosphere, and that makes sense actually. That is expected, as you expect more diversity in the bulk soil than in the rhizosphere, than much more in the roots, as the plants tend to recruit what is more beneficial to them. Then looking at the community composition, they were also different. So the rhizosphere and roots were different. Because of this, I presented the two compartments separately. So this is the community composition for looking at the products that were applied in the feed and how they impact the microbiome in the greenhouse experiments. So each dot again represents the bacterial community of one replicate sample. You can see the control different from every other thing. So this is for the rhizosphere compartments. Then in the root compartment, we can still see some level of separation where the organic acid one was different from everything else. And also this, it's explaining up to 18% of variation is explained by the soil treatment or the biological product treatment effect on the bacterial communities. But then again, I wanted to see differentially abundant taza. Since the communities are different, are they microbes that actually have higher abundance with respect to the treatment that was applied in the field? Then I found here, you can see the x-axis showing the biological products, while the y-axis is showing the different taza. So the purple represents significant reduction, while red represents increase, and the darker the color, the higher the more significant the reduction or increase. So I found some well-known beneficial microbes like Steroidobacter and Actinophytocola that are known to produce secondary metabolites and even antibiotics to be higher in plots that receive the biological products compared to the baseline control. This is in the rhizosphere. Then in the roots, I also found very interesting ones like Lysobacter, significantly higher for plots that receive the fish and seaweed products compared to the control. Then also Caulobacter is a well-known plant growth promoter, was higher for those that receive the products compared to the control, which means they may be contributing to reducing nematode infestation. Then I wanted to see what is the relationship between this bacteria taza and symptom rating. So this is showing like Spearman correlation between the relative abundance of the taza and symptom rating. We can see the gray shaded area represents the confidence interval and 5% confidence interval. We can see that the higher the abundance of the microbes, the lower the symptom rating. This is true for Steroidobacter and other beneficial ones such as Actinophytocola, Caulobacter, and Lysobacter. So in conclusion, actually all biological products increase the abundance of certain beneficial and antagonistic bacteria to reduce nematode infestation. So these microbes then can now be isolated and tested for potential agro-system applications, which means we need to isolate the microbes and test in more controlled conditions to validate the findings and know what the microbes can really do. Then results also suggest that if we apply biological products in infested feeds, it may precondition the soil to reduce nematode infestation in the following cropping season. I would like to acknowledge the industry partners and project partners for their support in this work. And I would like to thank Murdoch University for postgraduate scholarship. And also I would like to thank my supervisors for their guidance and the tomato growers for giving us access to conduct the field trials, the property. Then my colleagues that assisted me with field work and also nematology unit for their guidance with the nematology aspect of my work. Yeah, thank you everyone for listening and happy to take questions. Thank you.
Okay, thank you, Manuel and Henry. So we are now going to start the Q&A section. So I've got the Q&As over here. Again, if you see a question you want to have answered, upvote it because I'm going to start from the top and go down with a little bit of jumping back and forth between the two speakers. So first question we have, and I would ask speakers, please try to make nice, succinct answers because we've got a lot of questions here and we're probably not going to be able to get through them all. So, Manuel, how long after inoculation did you measure protein expression? And for how long can bacterial strains, that's a lot of stuff. So how long after inoculation did you measure protein expression? And for how long can they keep colonizing it?
Oh, okay, that's a good question. So we grew our plants for 14 days. So we inoculated right at the beginning and grew for 14 days. We have done more longer term studies that the colonization pattern definitely changes, which microbe is abundant, changes over time. But yeah, we worked with 14 days.
Okay, and the second half of that is basically like how long can they keep colonizing / what influences that factor?
Okay, so I have to speculate a little bit. That's actually a study we are doing now. The idea generally in the field is that microbes continuously colonize the growing roots, right? A root grows, we have the root tip, it keeps expanding. And that's actually the zone of initial and major colonization. So I would say as long as the root grows, colonization is happening. But that's definitely something we're looking into by actually measuring proteomes of specific root portions now.
Okay, and then next question, also for Manuel. So do the mutants show that the type 6 secretion systems are important to colonization under non-notobiotic systems as well as notobiotic systems?
We have not tested the mutants in natural soils. What usually happens, and I think that's a lot of people working towards it, is what usually happens if you're doing single inoculations on plants in native soils, usually your inoculant gets outcompeted by what's naturally there. So we have not tested that yet.
Okay. All right, and then for Henry, how long did each of your trials take? And I think based on the timestamp, this is specifically asking about the first half of your talk, with the comment that it's rare to find a change in a fungal community in a short period of time. So how long did that initial trial last?
So the trials were conducted in different seasons. It's actually within the summer. It's 4 to 5 months. But the second season lasted longer, up to 6 months, because it was like elongated summer. Yeah.
Okay. Back to Manuel. So, okay, so someone working on another notobiotic system, they think that the most up-to-date one for larger plants is EcoFab 3.0. They're wondering if your system has any unique features or advantages compared to that one, to the EcoFab 3.0, if you're familiar with that other one.
I have looked briefly at the EcoFab 3.1. I don't remember if it includes, for example, the ability for drainage. Also, I'm not sure what the size limits are for upwards size limits, so I cannot really speak to that. I would have to look at that again.
Okay, and then it seems like related to that, next one is someone asking you just tell a little bit more about the notobiotic system that you're developing. So it looks very cool with the glass and everything. Can you give a bit more detail on it?
Yes, so we, and in addition to making it high enough throughput, we're also trying to make it affordable. So the cylinders are actually very cheap. You can buy them on Amazon. They're like if you have candles outdoors for a romantic dinner, you can have these cylinders. They cost like $100 for like 10 cylinders. So that's a very cheap component. The top is covered by a filter gauze so that we can actually reduce the humidity, because in the antibiotic systems, often the humidity is the main issue, like humidity buildup in the system. So we have like constant flow of air, but it's all through filtered. And the bottom, the key thing that we had to develop was really like the holder that keeps it stable and allows us to assemble everything. So initially we did this all with 3D printing and we did multiple iterations. once we had a system that worked, we actually then contracted to a company to make that on a large scale. So we bought like a lot of these plastic things that hold the cylinders.
Okay. All right, back to Henry. Oh, it jumped around. Here we go. So what will you be doing after the success of the tomato crops? Will you be trying any other plants and or commercializing any strains?
Yeah, I guess that is the next stage of the work. Doing isolations, like we are like there's next phase of the trial, the project, where we are doing some isolation of the microbes, and we are going to be testing them. If we see anyone that is very interesting, then commercialization can be in the next step, but we need to do isolation first. Then, yeah, so we are doing isolation of the microbes, and some of them we are going to be testing for the ability to promote plant growth, tomato growth. So if there's anyone that is very interesting and maybe unique or novel, there may be need for commercialization. Yeah.
Okay.
But first we need to isolate, yeah, and test. Yeah.
Okay, and then, so I think this is back for Manuel. How do you discriminate proteins secreted from the host and from the bacteria?
Oh, interesting. So we use this high resolution mass spectrometry approach where really we measure the masses of intact peptides as well as the fragments of those peptides. So, after measuring the intact mass, we smash them up in a gas and then measure the fragments. Now, plant and bacterial sequences do differ quite a lot in terms of amino acids, right? Even if it's the same protein, like a homolog, it's still very different in sequence. Each amino acid that is exchanged actually completely changes the mass of the peptide. So with our approach, we can very clearly differentiate between this is the peptide with this sequence, not the peptide with this sequence, that where the one amino acid difference. Now, so between plants and bacteria, we have no issue discriminating. There's, if you have some very closely related strains, we tried, for example, with 2 strains of Rhizobium leguminosarum at some point, there it gets more tricky because there's so much sequence similarity that it's hard to discriminate. But generally, there's no issue based on mass to discriminate between plant and microbial proteins.
Okay, then Henry, let's see. So Henry, you said that in your, again, I think this is your first trial, you said that Farm B was non-conventional, and that seemed to have a larger effect of the products that you applied in that case. Can you go into more detail about what non-conventional meant and what sort of farming practices you speculate might be involved in that different effect?
Yeah, with the non-conventional farm, there was no synthetic fertilizer applied and no herbicide or even pesticide. There was just no synthetic inputs. So probably that's why I found higher response from the biological products, which means if we reduce the use of synthetics and increase the use of biologicals, do we expect more effects compared to when we are mixing them together? Yeah.
All right, and let's see, back to Manuel. So some notice in the PCA plot of your proteomes that the microbial inoculation spots seem to have 2 distinct groups. It's like, are there any sort of phylogenetic taxonomic relationships among the bacteria that form those 2 groups? And I think this is referring to when you showed how the plant was responding to the microbes.
Right. So 6 of the microbes are gram-negatives. 1 of them is gram-positive. The gram-positive is definitely one that kind of stands out separate from everything else. For the other ones, they're all proteobacteria. Well, most of them are proteobacteria. So nothing specifically stands out. But of course, we can assume that there's very big differences in surface structures and flagella and things like that that the plant might actually be sensing. And of course, the metabolites and whatever things they put out might be differing a lot. But the gram-positive definitely leads to a completely different host expression pattern.
Henry, regarding your nematodes, how many of them did you actually inoculate as the starting egg count?
How many what?
How many nematodes did you inoculate with the starting egg count when you were testing the nematode
Yeah,
analysis?
with the inoculation, I inoculated the plants using freshly extracted eggs of Meloid organic javanica, approximately 10,000 eggs per pot. Yeah.
Right. I see multiple questions asking about open positions. I'd encourage you to contact the speakers about that afterwards on your own. Meanwhile, Manuel, how do you make sure that there are no microbes in the notobiotic systems? And this is what I'm going to put my own thumbs up on as well, because I've never managed to find maize that doesn't have some endophyte still living inside it somehow.
Yeah, that's the crux of it, right? And we actually published a paper on sterilization methods for corn seeds a while ago. A lot of corn seeds are really full of all kinds of fungi and bacteria. So we developed different protocols, of course, surface sterilization with ethanol and bleach. Some people add antibiotics or fungicides to the mix during germination, which also helps a lot. An additional treatment that we have found helps a lot is to heat the seeds. I think it's 50 degrees Celsius overnight. So that's described in the paper. So heating kills a lot of less seeds ultimately germinate, but you also really get rid of a lot of the internal stuff. But you will always deal with a little bit of contamination. So you definitely have to watch out for that and make sure you control for it.
Okay, thank you. All right. Henry, so you didn't see any strong community level changes from your treatments, but do you have any plans for using some sort of additional technique that says modeling techniques here, like structural equation modeling, to try to figure out the difference between specific microbial taxa and specific treatments that might have increased yields? Basically, do you have any plans for starting to tease apart that?
Yeah, I did look at the abundance of those microbes from the first trial. I didn't do structural equation model, but I did look at correlation because I didn't present that one here, but I found some of them were positively correlated with yield. Some of them positively correlated with biomass, which suggests they may be contributing, but then it's still hard to conclude because that is just based on model or correlation. So there's still need for extraction, isolation of the microbes and testing them in more controlled conditions to validate that finding. Yeah.
Then I think this may be our last question. So Manuel, so is there any work being done on improving the medium for the in vitro growth, since you had all those genes being different from in vitro versus in vivo? Or do you think that that medium is actually capturing enough to be sufficient for your model?
I'm not quite sure what the question would refer to. So what we tried with this medium was to minimize. So, we started with the MS medium for plants, and we tried to add enough things that all the microbes can grow, but not more. There might be chances that we could actually reduce for specific microbes, take out a few of the compounds to make it even more defined and simplistic. That's definitely an option. We haven't started looking in that because we want to be able to just grow all 7 microbes with the same medium. But kind of that's the extent of what we were going for.
All right. Okay, thank you very much. So we are now at our 1 hour mark. So thank you, everyone. If you have questions that we were not able to get to, I'm sorry. The contact information for our speakers is in the chat, so you're welcome to send them an email afterwards to get in contact with them with additional questions. Thank you very much for attending our webinar, and we hope we see you in future webinars from the Phytobiomes Alliance or even at our workshops at PAG. Take care, and y'all have a great week.